Hi guys, I'm Manya Patel, and I'll be going over transcription for Unit 6 of AP Bio. First of all, what is transcription? Transcription is the synthesis of RNA using information in the DNA, and it produces mRNA. In eukaryotic cells, this happens in the nucleus, and in prokaryotic cells, it happens in the cytoplasm. So there are three main phases that happens during transcription: initiation, elongation, and termination. So for the first phase, which is initiation, an enzyme called the RNA polymerase II separates DNA strand and joins the complementary RNA strand nucleotides with it. The RNA polymerase binds to a specific DNA sequence called the promoter, and this promoter includes a TATA box in eukaryotic cells. And this box indicates where a genetic sequence can be read and decoded. During this, the DNA's double helix starts to unwind and the two strands separate, and one of the strands becomes the template strand. On the image on the left, you can see the promoter region and how the RNA strand starts to come and unwinds the double helix. Some key vocab words to remember during initiation is RNA polymerase, which synthesizes DNA molecules from a template of DNA, and you don't need a primer. A promoter, which is a specific region of the DNA located upstream of a 5' gene. And the TATA box, which is a binding site for transcription factors and for RNA polymerase to initiate transcription. The next stage is elongation. In elongation, the RNA polymerase moves along the template strand from a 3' to 5' direction. It starts to build and to synthesize the RNA strand in the 5' to 3' direction, and you can see in the image on the left how this kind of works. You can see the nucleotides start to come, and it's still unwinding the double helix. And just so you can differentiate between DNA and RNA and the bases, for RNA, it's just a single strand, and there are four main bases for this. There's cytosine, guanine, adenine, and uracil, which is slightly different to DNA. In this image, you can see the DNA has almost the same basis, except slightly different. It has cytosine, guanine, adenine, and thymine instead of uracil. DNA is also the double helix formation, where RNA is only a single strand. And just so we can get a closer look at this, for DNA, a nucleotide includes a base, a sugar, and a phosphate. And how these are paired is the adenine goes with the thymine, the cytosine goes with the guanine, and vice versa. Lastly, we have termination, which in this case, the RNA polymerase reaches a stop sequence. The RNA strand is then released, and the DNA strands rewind back into a double helix. And the product that is formed is the pre-mRNA for eukaryotes. The pre-mRNA is the immediate unprocessed transcript produced from the DNA by the RNA polymerase during transcription. So it's basically just the initial products formed, and you can see the whole process better in the picture on the left. Lastly, before the mRNA leaves the nucleus for translation, there are three main things that occurs: the addition of the 5', addition of the poly A tail, and splicing. So for the addition of the 5', a modified guanine nucleotide is added to the beginning of the RNA strand, and this is called the 5' cap, and this basically helps protect the mRNA. You can see this better on the picture on the left. Next, we have the addition of the poly A cap, and this is basically a long chain of adenine nucleotides, which is added to the end of the RNA strand. And what this does is that it binds special proteins and protects the RNA for translation, and it strengthens efficiency. You can see this better in this picture, where you can see the poly A cap, which is like the nucleotides, added to the end of the pre-mRNA. And lastly, we have splicing, which is one of the most important factors of this whole process. For the pre-mRNA sections are called introns and exons. Introns are the non-coding regions, and exons are the coding regions. And during splicing, we remove the introns, and they join the exons together. For the whole process of splicing, a spliceosome, which is inside the nucleus, performs the RNA splicing. And this is a necessary feature of splicing. The spliceosome cuts the introns out and connects the exons together, and this will result in a continuous coding sequence and helps the RNA mature. You can see it's better in this picture where you can see the spliceosome cutting out the introns and it joins the exons together. What if there were no spliceosomes during transcription? If there are no spliceosomes, then the introns would still be in, and this will cause the protein that is produced to be incorrect and the gene will not function properly. So that's all for transcription. Thank you guys so much for watching.